Hello, and first I want to thank the organizing committee for letting me presenting this work. It's about the upper and lower respiratory tract microbiome in calves, interaction and dysbiosis during acute bronchopneumonia. So, as we already know, the bovine respiratory disease complex is a major economic and public health burden affecting young bovine worldwide. It is a multifactorial disease that involves the presence of one or at least several viruses and or bacteria at the same time. And these respiratory pathogens act in the presence of a very complex network of resident bacterial species called the microbiota. And many disturbances can lead to a state of a disturbed microbiota called dysbiosis. So, historically, we moved from considering the lungs as a sterile organ to recognizing the presence of a resonant and a normal microbiota in the lungs. And now, actually, we think that there might be some sort of interaction between the members of the microbiota itself and between this microbiota and the infecting pathogens. So, the goals of this study: first, we wanted to describe the complex structure of the upper and lower respiratory tract microbiota during health and during the bronchopneumonia disease. Second, it was to determine if well-known respiratory pathogens have an impact on the upper or lower respiratory tract microbiota structure. For that matter, we sampled 70 calves, 2 to 8 weeks old and having acute signs of bronchopneumonia. These calves were selected from 23 breeding farms in the south of France. In parallel, and as a control, we selected 21 calves, healthy calves, same age, and from the same region in France also. These calves were clinically healthy. From every calf selected, we took nasal swabs to describe the upper respiratory tract and bronchoalveolar lavage for the description of the lower respiratory tract. On these samples, so the nasal swabs, simplified by NS, and bronchoalveolar lavage, BAL, on these samples, we performed RT-qPCR specific for the classic respiratory pathogens, so the BRSV, bovine respiratory syncytial virus, the bovine parainfluenza type 3, the bovine coronavirus, Manemia hemolytica, Pasteurella maltosida, Histophilus somni, and Mycoplasma bovis. In addition, on the same samples also, on all the samples, we performed NGS sequencing, so deep sequencing on every sample, in order to perform microbial profiling of the microbiota and to describe if there is any pattern of association and interaction using several methods, several regression methods. So this is a scheme representing what we have done in the big study, but now we are presenting just this one. So NS and BAL samples, the microbiota, we're using a specific targeted actually amplicon deep sequencing targeting the 16S RNA gene, doing the microbial profiling and the interaction study. In parallel, we also did virome study. We will not present it due to lack of time. So sequencing was done on a Illumina HiSeq platform. As a first result, we noticed that the microbial richness in the upper respiratory tract, in the nasal swabs, was significantly higher in both groups of calves, so in bronchopneumonia and the clinically healthy calves, compared to the microbial richness of BALs. As we can see in this illustration here, doing several tests of richness, where the red dots describing the upper respiratory tract and the blue dots describing the BALs, you can see that in every metric calculated, you can see that the richness is really higher, significantly higher in the upper respiratory tract. The second interesting result we have found is that there was a clear dissimilarity or disparity between the upper and lower respiratory tract and the bronchopneumonia group. This disparity is based on two things. First, of course, on the abundance of the communities detected, and second, on the taxonomy, so on the identity of the microbiota itself. This illustration we can represent on the x-axis the IDs of the calves, and horizontally it's the microbial structure of every calf. So each color represents a phylum. This is at the phylum level. The first half of this illustration is the nasal swabs, and the second half is the bulbs. As we can see, it's really different, just looking at the colors here. And for example, just describe the, just going to talk about later. So, the same disparity, you can see it also when plotting these results. So every dot represents the microbiota of one calf. As you can see, the blue, the bars, and the red are the NS. The same disparity was also noticed in the healthy calves, but very less prominent. So we can maybe say that a taxonomical disparity comes during bronchopneumonia. Here we listed the most abundant component of these microbiota. First, and the NS of the bronchopneumonial calves, and here the bulbs from the lower respiratory tract. And the only thing in common was the really high abundance of the Mycoplasma genus. The only common thing. As for the next of our study goals, that means searching for interactions or association between the microbiota and an infecting respiratory pathogens, known respiratory pathogens. So one of the 7 detected by RT-PCR, we did an exploratory analysis using PLSDA and SPLSDA regression mode methods. And I really simplified the result. Like here, we found several patterns of interaction and association. For example, Mycoplasma bovis was tightly associated with Pastorella maltosida and less tightly with another group of bacteria. The other association, for example, the species Histophilus somni was also tightly associated with several genera like Plonomicrobia, Microbium, and the 3 genera from the Pasteurellacea family, like the genus Histophilus, genus Pastorella, and the genus Manemia. As for Manemia hemolytica, it was associated with another genus called Murdochiella. As for the respiratory viruses, they were associated always together. Furthermore, we search for an impact of presence of one of the 3 viruses to the structure of this microbiota. Of course, during bronchopneumonia, since we're talking about the presence of a virus. And for example, we noticed that the presence of a virus is associated with a higher abundance of Mycoplasma genus, whereas the opposite was noticed during the absence of a virus for another group of bacteria, especially Manemia and Truperella genera. And here also not only Mycoplasma, but also another group of bacteria. So as a take-home message, we are dealing with a really diverse microbiome with a clear, distinct structure observed between the upper and lower respiratory tract. And now the questions asked, what are the factors affecting this microbiome? Is it the environment, the breed, the age, the time of the sampling, geographic. We are trying to answer these questions. The genus Mycoplasma was really omnipresent in both clinically and healthy affected groups in both respiratory sites. As for these pattern relationships we saw, we must search for the mechanism of interactions affecting these association and interactions. And what we did here is really trying to develop a model. And for the future, as a future perspective, we will use the virome data in order to make the same study. Thank you for your attention.